and I'm the principal investigator of the Moose Genome Project. Moose are an interesting and important species to study. Biologists believe that large mammals like moose will be the first impacted and perhaps the most severely affected by climate change. Days like today make explaining the importance of my research pretty easy. Today is January 10th, and as you can see, it's quite rainy here in Peterborough. The temperature is about 6 degrees Celsius, despite the historical average being around -4. This means that even in January, moose are experiencing heat stress. This is because in their winter coats, temperatures of about -4 degrees Celsius raises their metabolism, which burns necessary fat, causing moose to eat far more often. And in winter, this is really important because highly nutritious foods are usually scarce, and malnourished moose can become easy prey for predators of the north. My project is looking to identify the genetic component that may be tied to the ability of moose to adapt to these changing conditions that we see today, particularly in regards to climate. Now I'm here at Trent University, standing outside the DNA building where all of this research takes place. After traveling and collecting samples all across Ontario, I'm currently working inside the genomics lab to gather and analyze data. Let's take a look. Gathering data in a genomics project takes time. The longest part is the waiting game of collecting samples. I spent much of 2015 traveling to Ontario Federation of Angler and Hunter Zone meetings to give talks and collect samples. Kits like these are handed out so that hunters can submit small pieces of meat, which are sent here. Once a kit is received, sample preparation begins, where DNA is extracted. The DNA is then put into a special plate and undergoes amplification. This DNA then undergoes amplification by polymerase chain reaction. It uses temperature cycles to stimulate DNA replication, so a few copies of DNA can become millions in a matter of hours. We only select specific areas of DNA to amplify, rather than all of the DNA in the sample. These specific areas are called loci and reveal different versions of important genes. And these genes are all in some way tied to climate change. The goal of the project is to identify variation within these genes that could infer differential selection by environment. I expect to find such variation because my previous work has identified that there are actually 2 different subspecies or types of moose in Ontario. Subspecies were formed during the last ice age, where groups of moose were separated in different refugia. This means that these different types had at least 10,000 years of separation, during which they would have evolved differently according to their past environments. As ice sheets receded, moose would have shifted their ranges further north, but this is a slow process. In fact, these 2 subspecies have only met up quite recently, as early as the late 1800s. To identify the genetic component of adaptation, we are first looking for genes related to climate that are variable. This variation implies that selection may have acted upon the gene in some way. Interestingly, the genes we are looking at contain a repeat region that is particularly mutable or more likely to change when under stress. For example, a heat shock protein gene, which functions in heat tolerance, is regulated by an HSP protein, which in normal functioning stabilizes the DNA fragment during replication. This allows for an exact copy to be made of the DNA, resulting in the same level of heat tolerance. However, if this individual undergoes heat stress, the protein disassociates from the DNA so that it is no longer stabilized. This allows for slippage to occur during DNA replication, which can result in incorporation of a new segment or an additional repeat within the region. This would mean that a new trait, or perhaps a greater heat tolerance, could result. This means that as moose are exposed to heat, they would likely be able to adapt to it. Now this is hypothetical, but identifying a gene like this would mean that we have the capacity to understand the ability of moose populations to adapt to changing conditions as temperatures warm. Currently, this project is still in the data generation stage. It's expected to be completed by the end of February. The methods utilized in this research will also be applied to moose in Manitoba, where we have a unique temporal data set to investigate how moose and their environments have changed within 20 years. Wildlife Conservation Society Canada and the W. Garfield Weston Foundation's fellowship has allowed me to expand my research to survey a dozen genes instead of just 5. With a candidate gene approach, as utilized in my research, Having a large number of genes increases the chances of identifying important genes or gene regions. So I would like to thank the W. Garfield Weston Foundation for their support of my research, which has helped me develop my work into a more comprehensive survey. Are we starting? Okay. Yep, can I start now? Does that look like I'm looking down?
Okay, go away and then come back.
Can I come? The Foundation's fellowship has allowed me to expand my research to a dozen zines. My mouth is so frozen.